sharing the facilities of the School of Biotechnology in the form of hands-on training. School of Biotechnology is an autonomous university teaching department, Rajiv Gandhi Pradyogiki Vishwavidyalaya, the University of Technology of Madhya Pradesh, accredited with grade A by NIC, Bhopal. Therefore, SOBT, abbreviation of School of Biotechnology, is organizing Consortium Biotech Workshop, which is a series of 5 workshops on the core sectors of biotechnology and its allied fields. These workshops are as follows. Workshop 1, how to start the lab work under aseptic conditions, which was already done on 1st and 2nd of June 2020. Workshop 2, how microbes are fascinating, which we are organizing today and tomorrow, that is on 7th and 8th of June 2020. Workshop 3 is focusing on how to study the beauty of DNA, that is to be organized on 14th and 15th of June 2020. Workshop 4, how the gene is expressed in the form of protein, that is to be held on 21st and 22nd of June 2020. Last but not the least, for workshop 5, how computer helps in the designing of biological data, that is to be held on 28th and 29th of June 2020. We hope you have received your first workshop e-certificates, School of Biotechnology information brochure, and the syllabus of the courses running in the School of Biotechnology, RGPV Bhopal, that is MTech Biotechnology, and PGCMB, that is Postgraduate Certificate Program in Medical Biotechnology. A fresh request to all the participants, those who are interested in real sense for the hands-on training, register themselves on the link given below on the e-certificate. Copy that link and register yourself for the respective workshop. Today, we are focusing on the workshop, that is, now this is the time to Shankanath for the today's workshop, how the microbes are fascinating. This today's workshop has been divided into 4 sessions. Session 1 contains microbial strain selection and their improvement techniques. Session 2 comprises of media, broth selection and their preparation, cereal dilution, plate preparation. These topics we will deal today in the 2 sessions, that is first and second. Tomorrow we will deal in the third and fourth session. In third session, slant preparation, streaking and spreading, growth curve analysis of bacteria, and in the fourth session, that is the very valuable session, that is only on the basis of your feedback. That is feedback session. Welcome to the fascinating world of microbes. Whenever we are designing our experiments, there is a need of any model system. Whether we are planning for the lab work, that is lab experimentations as a routine classwork, or we can say that whenever we are designing any project work or any of the type of experiments, there is a need of model system. Then only it is possible to perform our experiments. Now, today's workshop's first session is based on microbial strain selection and their improvement techniques. Whenever we are talking about the microbial strain, for instance, we have selected the Escherichia coli. Escherichia is the genus, coli is the species, and this is the strain of this is the Escherichia coli. These are the 2 strains of Escherichia coli, that is DH5 alpha and DH10 alpha. But strains are not having the proper position in the classification, whether it is a bacterial classification or it is a fungal classification or it is plants, animals, humans, etc., etc. The reason behind this is
Excuse me, ma'am.
yes, please,
Ma'am, some students are waiting for let them in, so you can take them, please.
sure, please.
Sorry, ma'am,
But
for disturbance.
here nobody is waiting.
Yes, ma'am.
Here nobody is waiting.
Ma'am, they are saying that it is not taking them in.
No, no, no. There is no. Once anybody is coming, na, some beep is there.
Okay, ma'am. Sorry.
Shall we wait for them or shall we start?
Ma'am, I have tell them to again invite, so they are doing.
Sure. We are just waiting for 5 minutes. Just
Okay, ma'am.
request them, please, if they want, they please join us.
Okay, ma'am.
I think nobody is trying to enter. Shall we continue?
Yes, ma'am.
Okay, okay, ma'am.
Whether they are coming or not?
They are saying they are trying, but it's not working.
They're having the link.
Yes, ma'am.
You just say kindly disconnect themselves first, then
Yes,
reconnect.
ma'am. Okay, ma'am.
Okay?
Okay, ma'am.
I think we have to start. We have to continue, na?
Okay, ma'am.
Is it visible to everybody?
Yes, ma'am.
We are talking about the position of strain in the classification, whether that is the classification of microbes or the classification of plants or the classification of animal. Genus is there, species is there, but there is no permanent place, or we can say that category of strains. The reason behind this is because among these, this is the genus, and that is genus species. The Escherichia coli is the genus with species, And these two are the strains. All these three are gram-negative bacteria, bacillus, rod-shaped, but this DH5 alpha is having the special characteristics which are not present in Escherichia coli as a wild type and the DH10 alpha. Similarly, this DH10 alpha is having the specific characteristic which is not present in DH5 alpha or the Escherichia coli. Only very minor difference among these, or we can say that only on the basis of that special characteristics, these are differentiated in various types of strains. Escherichia coli, then the strain as DH5 alpha, DH10 alpha, and so on. Hence, we can say that the strain is a genetic variant or subtype of a microorganism, plant, or rodents. They're having very specific characteristic, very minor difference. That's why there is not having the enough space in the classification of any of the types. When we are talking about the strain, on what objectives we have to select our strains? What are the basis of selection of strains? The first and the foremost important thing whenever we are selecting the strain is that strain should have to improve their technological properties. I'm repeating, microbial strains for the manipulation, they have to improve their technological properties. Which type of technological properties? Again, question arises. Two important technological properties. Number one is to improve product yield, and the second one is to improve growth characteristics. Number one, to improve product yield, and second is to improve product characteristics, on the basis of which we have to select our strain for our experimentation. Or we can say that strains are selected on the basis of these two important factors: to improve product yield, to improve growth characteristics through manipulation. Now, what are the various sources of the strain from where we will collect all these? In our environment, n number of microbes are present, but these are not having the characteristics of the strains which they are having. Almost all the strains are man-made or artificial strains prepared in the lab itself, that is under in vitro condition. Whenever we are talking about the artificial strains which are prepared under lab conditions, they are mainly available in the various institutions that are considered as the repositories of these microbes, or we can say that the organisms. One is available in India, that is MTech Chandigarh, for the collection of the microbial strains. Apart from this, ATCC is also there in USA. Once we are designing our experiments, we are mainly preferring to work in the simplest manner in the case of microbes. Then again question arises, this question is growing in our mind, why microbes are preferred to plants and animals? Over such characteristics, these microbes are having the two important reasons. The first one is generally cheaper to produce, and second is enzyme contents are more predictable and controllable. First, cheaper to produce, and second is enzyme contents are more predictable and controllable over the plants and animal tissues that contains more potentially harmful materials than microbes. The best example is phenolic compounds, that is from plants. Now, whatever the strain we have selected, and whatever the product we are desiring, that strain is not in a position to provide that desired amount of product or desired result. And there is no other strain which is available here to give us such type of desired product or desired result. Then there is a need of strain improvement. Why we are improving the strain? Again, to enhance their metabolic capabilities. Why enhancing of the metabolic capabilities? For biotechnological applications, with the manipulation and improvement of microbial strains. Only one motto behind this, why we are improving the strain. The reason behind this is to enhance their metabolic capabilities. Why we are enhancing? For the biotechnological applications, how we are enhancing? With the manipulation and improvement of microbial strains. What is the purpose of strain development?
Sorry to interrupt, ma'am, but your slide is not visible.
Slide is not visible.
No, ma'am.
Ma'am, someone is presenting.
बलजिंदर सिंह प्लीज अपना प्रेजेंटेशन बंद करिए। आप बीच में स्लाइड प्रेजेंट ना करें प्लीज, इंटरप्शन होता है। वेरी सॉरी मैम, प्लीज मैम अब आप कर रही है मैम। बीच में कोई भी स्लाइड प्रेजेंट मत करिएगा प्लीज।
Shall we start from here or back?
फ्रॉम हियर मैम, चलो।
From here
यस मैम,
or from here?
फ्रॉम बैक स्लाइड मैम।
This one?
यस।
Why we are just improving the strains? What is the need for the improvement of strain? The reason behind this is to enhance their metabolic capabilities. Why we have to enhance their metabolic capabilities? Again, for biotechnological applications, how we are enhancing or why we are enhancing such type of techniques for biotechnological applications, that is with the manipulation and improvement of microbial strains. Now, what is the purpose of strain development? First, we have selected the strain. What is the objective behind the selection of what is the basis behind the selection of the strain? That is clear. Then, why we are improving the strain? Then, what is the purpose of strain development? N number of purpose to develop a strain. Number one, to relate individual cases to an outbreak of infectious disease. Nowadays, the best example is novel coronavirus and the disease is COVID-19. Another purpose is to establish an association between an outbreak of food poisoning and a specific food vehicle. Next one is to study variation in the pathogenicity, virulence, and antibiotic resistance of individual strains within a species. Other purposes are to trace the source of contaminants within a manufacturing process. Another one is to study the microbial ecology of complex communities such as biofilms. And one more purpose for the strain development is to characterize microorganisms with important industrial applications. Now, the purpose is fixed. This is the purpose. For this purpose, such type of strain is required, but that strain is not in a position or it is not capable to produce our desired amount of product. There is a need of strain improvement. Now we have improved the strain. Then what are the targets of strain improvement? For the rapid growth, for genetic stability, reduction of cultivation cost, lower price in nutrition, lower requirement of oxygen, production of and additional enzymes, compounds to inhibit contaminant microorganisms. Along with this, that target should be non-toxic to humans. Large cell size for easy removal from the culture fluid, ability to use cheaper substrates, elimination of the production of compounds interfere with downstream processing, increase productivity to improve the use of carbon and nitrogen sources. These all are the targets of strain improvement. Now, strain is decided, basis of strain has already been selected, then improvement of strain was selected, purpose of improvement, targets has been decided. Now, what are the various methods for strain selection? Some Examples of the methods for the strain selection are biotyping, bacteriocin typing, protein typing, phage typing. Biotyping. The strains which are using for the biotyping which are considered as the biotypes. For instance, we have selected DH5 alpha and DH10 alpha for the study of the differences in the biochemical reactions between these two strains of bacteria. That is, one strain is DH5 alpha of Escherichia coli and another strain is DH10 alpha of Escherichia coli. These two strains are considered as the biotypes. And our main motto, why we are doing the biotyping? Just to analyze, just to study the differences in the biochemical reactions between these two strains of any microbe. This is known as biotyping. And what is bacteriocin typing? Bacteriocin, CIN, that is bacterial toxins typing. Or we can say that antibacterial products, typing of antibacterial products. Protein typing, as its name suggests, typing of the proteins synthesized by the different strains of the microbes. Which type of protein is present? What is the purpose of that protein? In which manner that protein works? These all are included under protein typing. Phage typing, that is sensitivity of the test strain to the lytic action of standard phases. So these are some representative methods for strain selection. Now, strain is selected on the basis of the requirement of the experiment. The purpose of strain has been decided. The targets has been decided. There is a need. If there is a need, then the improvement of the strain should be done as per the requirement of the experiment. Now, optimization of microbial activity. Three important characteristics can be optimized for the microbial activity. First is optimizing environmental conditions. Second one is optimizing nutrition of microorganisms. And third is other includes methods not involving foreign DNA, that is mutagenesis. And another method is methods involving foreign DNA, that is DNA recombination. These are the three important categories where we are optimizing the microbial activity. The first one is optimizing environmental conditions. Second one is optimizing nutrition of microorganisms. And the third one is other includes methods not involving foreign DNA. That is the method of mutagenesis and the methods, excuse me, methods involving foreign DNA, that is the process of DNA recombination. Whenever we are talking about the optimizing of environmental conditions, optimization of, that is modification of physical parameters, particularly the temperature, agitation, etc., which is required for the growth and development of microbes uniformly, homogenetically. Similarly, the modification of chemical parameters, pH, oxygen concentration, and modification of biological parameters such as enzymes. Need of optimization of such type of environmental conditions. Then optimization of nutrition of microorganisms in the form of carbon sources, mineral sources, nitrogen sources, precursors, enzymes. These are the 5 categories where we have to optimize the nutrition of microorganism, which is required in which quantity, with which how much of the concentration for the proper growth, for the uniform growth, for the homogeneous growth of that microorganism in a particular vessel. Now, other methods mainly not involving foreign DNA, that is the process of mutagenesis. Mutation is the process which occurs in nature by the sudden change occurs in the sequence of nucleotide. Any of the changes occurs in the sequence of nucleotide. Then under lab condition or in vitro analysis or in vivo, process of treatment given to microorganism which will cause an improvement in the genotypic and phenotypic performances. Process of treatment given to the microorganisms which will cause an improvement in their genotypic and phenotypic performances, both morphological and at the genetic level. Which type of changes occurs? Which type of improvement is required? There are n number of ways by which we can do the process of mutagenesis without the involvement of foreign DNA. The process is deletion, duplication, inversion, insertion, translocation. This is our original DNA. Suppose the process of inversion takes place. Here in the original DNA, A, B, C, D, E, F. And here, by the process of inversion, A, B, C is replaced by E, and E is replaced by C. That is inversion. Here also, here. That is exchange of the segments, exchange of the fragments. Now, deletion. A, B, C from, this is the original one, and this is the process of the fragment of the DNA after the process of deletion. That is, C is deleted. There is no C, A, B, D, E, F. Here also, this fragment is deleted. Now, insertion, A, B, C, D, E, F. In between D and E, there is an addition of or insertion of L fragment. This fragment has to be inserted here. And duplication, that is, this is the place where the duplication of D fragment takes place. Their duplication not is present in the original DNA, but here under the process of duplication, this D is duplicated. And the translocation, A, B, C, D, E, F in the first, that is the mother strand, and its complementary, that is LMNOPQ. This DEF is translocated, and they are just translocated where? In the complementary strand, and OPQ is translocated in place of DEF. These are the various methods. Only changes, that is insertion, deletion, duplication, and changing of the positions, the expression of the gene is changed. There is no involvement of the foreign DNA. These processes are known as mutagenesis. Likewise, site-directed mutagenesis. We have taken the example of uracil deglycosidase deficient strain. Here, double-stranded DNA. Here, double-stranded DNA. For this uracil deglycosylase deficient strain, this T is replaced by U. Here, this T is replaced by U, and this double-stranded is converted into the single-stranded DNA. Likewise, point mutations. This is the original DNA, corresponds to the amino acid leucine UUA. And these are the various types of mutation caused in this simple original DNA having only one triplet, that is the triplet of amino acid leucine, UUA. Here, once the frame is shifting in between the U and A, and G is placed there, this is known as frame shift mutation. In between U and A, gene is placed. The frame is shifted in between the U and A, then that is known as frame shift mutation. Here, U is replaced by C silently, then this is known as silent mutation, UUA, CUA. This mainly represents leucine. Although the silent mutation occurs, but the leucine is coded. Then missense mutation, that is UUA is replaced by GUA, then that is known as valine. but we are looking for the amino acid leucine, corresponds to the amino acid leucine. That's why it is of missense mutation. And then U, this second U is replaced by A, then it is changed into UAA, considered under the category of nonsense mutation, that is denoted as stop codon. So these are the various places where at a particular point, the point mutation occurs without the insertion of any foreign DNA. Now, some mutagens which are mainly induced at the time of mutation, and what is the impact of that on the DNA, and which type of relative effect occurs when such type of mutagen is causing the mutation. Ionizing radiations, X-rays, gamma rays. Mutation induced, that is single or double stranded breakage of DNA takes place, and deletion or structural changes occurs, that is the impact on DNA, and relative effect is high. UV radiations, chemicals is the second category of mutagen. Mutation induced with the help of pyrimidine or dimerization of such type of bands. Then what will happen? Impact is on transversion, deletion, frame shift, transitions form. That is GC2AC. And the relative effect is medium. Similarly, the other mutagen, hydroxylamine, that is NH2OH, deamination of cytosine, and GC is converted into AT transitions, then the relative effect is low. N-methyl, N-nitroso-guanidine, that is methylation of bases and high pH, which mainly induces the mutation in the selected fragment of DNA. Impact on DNA, that is GC280, and transitions occur, and the relative effect is high. Nitrous acid, that is HNO2, deamination of AC and G, bidirectional transitions, deletion of AT to GC or vice versa, GC to AT. Relative effect is medium. Phage, plasmid, DNA transposing, base substitutions, breakage. Mutation occurs at this sites, either base substitution or the breakage takes place. Deletion, duplication, insertion, that is the impact on DNA, and the relative effect is high. Then ethyl methane sulfonate, alkylation of bases C and A, GC is converted to AT transition, and the relative effect is high. It means whenever we are selecting the mutagens, that selection should be based on the selection of the strains, selection of the targets, selection of the purpose. Hence, whenever we are exposing organisms to the mutagen, that should be in the haploid stage during the exposure. Why haploid? Why not diploid? Because many mutant genes are recessive in comparison to the parent or wild type gene. In the case of bacterial cells, these cells are haploid, while in fungi and actinomycetes, the haploid stage is found in the spores. So always, whenever you are exposing the organisms to the mutagen, the stage should be haploid stage. Now, mutagen is selected, stage is selected. On which stage we have to just provide the mutagen, that is in the haploid stage. Screening of microbial strains. How the screening of microbial strains takes place? That is with the help of random screening. The main motto behind this is survivors from the population are randomly picked and tested for their ability to produce the metabolite of interest or not. When we are focusing on the survivors from the population, a very large number of colonies must be tested. Then only the product of our interest, the metabolite of our interest is only produced, otherwise not up to the mark, product will be formed after the completion of all the steps of the experiments or the completion of all the experimental work. What are the advantages behind this? Why there is a need of large number of colonies? We are requiring the large number of colonies for the maximum production of our interested product or the metabolite of our interest from the survivors population. That's why minimal startup time is required. This is the first and the foremost important advantage. Minimal startup time is required. And second advantage is sustaining for years after years. But there is one disadvantage also, because random screening takes place, pick and choose method is applicable, then non-targeted and non-specific one. This is one and the only disadvantage of this technique. Only on the basis of random screening, non-targeted and non-specific is the disadvantage. That's why always prefer the rational screening. Rational screening means pinpointed. What is our motto? We have to focus on that. That is metabolic checkpoints. We must know about the metabolic checkpoints. Suggest ways to isolate mutants with specific traits. This is the strain. We have improved the strains. Purpose is fixed. Target is fixed. Method is fixed. Now, we just checking the metabolic checkpoints, with the help of which we are suggesting the ways to isolate the mutants with specific traits. Very specific. The chances of errors are very negligible. Another point, another factor which we have to consider under rational screening, environmental conditions, that is pH, temperature, aeration can be manipulated. As we discussed earlier, whenever we are selecting any strain, that is having the selection is on the basis of manipulation of the technological properties. Whatever is the environment required for the growth and development of any microbe, on the basis of the requirement of our desired product, their environmental conditions should be manipulated, such as pH, temperature, aeration. What are the various applications of such type of rational screening? A lot of applications. Now, some examples, for instance, selection of mutants resistant to the antibiotic produced. Second category of applications, selection of morphological variants, selective detoxification, selection of overproducers of biosynthetic precursors. These are some important categories of applications under rational screening. What are the screening tests for mutants? First, we have already decided this is our main motto, this is our objective. To fulfill this objective, this is the strain. This is the basis of selection of the strain, purpose of the selection of the strain, targets. If there is need, how to improve the strain also. We are having the specific methods and the specific reasons also for the strain improvement. Now, we are screening the test for mutants. Cells should be suitably diluted and plated out to yield 50 to 100 colonies per plate. Why we are doing this? Just to observe the morphology of the mutants or on some selectivity in the medium. Why morphology? Our main focus is to observe what are the changes developed phenotypically to observe the morphological behavior. Then, when morphological mutants are selected, the desired mutation may be pleiotropic. What is this pleiotropic mutation? In which change in one property is linked with a mutation in another character. There is a change in one property, but that is linked with the mutation in the another character. For instance, pleiotropic mutation is to be seen in the development of penicillin, yielding strains of Penicillium chrysogenum. This Penicillium chrysogenum, when after irradiation, strains of Penicillium chrysogenum with smaller colonies and were better producers of penicillin, and level of significance will be checked with the statistical tools. How much amount of penicillin is produced with the help of Penicillium chrysogenum? The result is around 2 to 3 folds more than which that was yield earlier. But whatever is the yield we are getting after manipulation, whether it is really showing the level of significance statistically or not, we have to check this also. Now, the another category, first, the category in which there is no involvement of the foreign DNA, in which we have discussed the various types of mutations with the help of the impact of various mutagens on the various strains of microbes for the specific purpose, either random screening or rational screening. What are the various methods which are suitable for such type of screening? Now, another category is methods involving foreign DNA recombination, that is on the basis of conjugation, transformation, transduction, and genetic engineering. Whenever we are talking about conjugation, this is the F plus cell of bacterium having the F. Why it is known as F plus cell? Because it is having the F plasmid, that is fertility plasmid, and this is the F negative cell, that is there is no F plasmid is present. These are the bacterial chromosomes. The bacterium which is having the F plasmid is considered as the donor cell, and the bacterium which is not having the F plasmid is considered as the recipient cell. And in between these two, there is a conjugation pilus. There is a tube which is known as conjugation pilus. This conjugation pilus joins these two cells together. F positive donor cell having the F plasmid and F negative recipient cell having no F plasmid, along with the bacterial chromosomes. Now, with the help of this conjugation pilus, the walls of both the bacterial cells are dissolved, and then one tube is formed. One gap is formed in between these two fused bacterial cells. These are showing the cells contact one another. Once the cell contact takes place, then there is a transfer of one fragment. There is a transfer of one fragment, or we can say that one strand of the double-stranded plasmid from the donor cell to the recipient cell. One strand of plasmid DNA transfers to the recipient. Once that strain is transferred from the donor to the recipient, what will happen? That recipient is making the complementary copy, and the donor is also making the same complementary copy of the one strand. Both the bacterial strains are separated, and this F positive cell is separated from the F negative, but not now. It is not the F negative. Now this F negative cell is converted into the F positive only because of the transfer of the foreign DNA to that bacterial cell to become F negative to F positive cell. This is the process of conjugation. What is the significance of such type of process? It is mainly playing an important role in the formation of some industrial products, particularly antibiotics. Another process is transformation. That is, foreign DNA is absorbed or integrated within the genome of donor cells. This is the DNA fragment from the donor cells, and this is the recipient cell having its own chromosomal DNA. Recipient cell takes up donor DNA. That donor DNA is inserted inside the bacterial cell with the help of electroporation. Once it is inserted inside the bacterial cells through electroporation, then the process of recombination occurs between the donor DNA and the recipient DNA. Whatever is the unrecombined DNA, that is degraded. Unrecombined DNA is degraded, and recombination occurs between donor DNA and recipient DNA. The remaining ones are degraded inside the cytoplasm itself. Now, this recipient cell has become genetically transformed cell after the incorporation of the foreign DNA from the donor cell to the recipient cell chromosome. DNA fragment from the donor cells, chromosomal DNA present in the recipient cell. Recipient cell takes up donor DNA from the outside, that is with the help of electroporation. And this, once this DNA is entered, then the recombination occurs between donor DNA and recipient DNA. Then this recipient cell has become the genetically transformed cell, and whatever is the uncombined DNA remaining, that was degraded. Cells in which transformation occurs that are considered as competent cells. Cells in which transformation occurs that are considered as competent cells. The method therefore has good industrial potential. Another method, that is transduction. Transfer of bacterial DNA from one bacterial cell to another by means of a bacteriophage, that is assembly of virus with the bacteria on a particular site is formed, that is phage infects bacterial cell. This is the first step of the generalized transduction. Then once the phage infects bacterial cells, host DNA is hydrolyzed into pieces and phage DNA and proteins are made. Once host DNA is hydrolyzed into pieces and phage DNA and proteins are made, then they are again reassembling and ready to infect the bacterial DNA fragments is packaged in a phage capsid. This is the phage capsid, which is hexagonal in shape. First, host DNA is hydrolyzed into the pieces and phage DNA has proteins and phage DNA and proteins are made inside this. They are having the two genes A positive and B positive. Here also A positive, B positive. Now, occasionally a bacterial DNA fragment is packaged in a phage capsid. This is the capsid, this one. Phage DNA is packaged inside this capsid having the A positive gene. Now the process of recombination takes place with the help of crossing over. When this crossing over takes place and both the strains or both the fragments are showing the process of crossing over, transducing phages infect new host cells where recombination, that is crossing over, occurs. Now we are having the recombinant bacteria having A positive gene. These recombinants have genotypes A positive, B negative, that are different from either the donor A positive, B positive or recipient A negative, B negative. It means it is a hybrid. Process of recombination takes place and one character is from here and another character is received from here. After the process of crossing over and recombinant bacteria is formed. The recombinants have genotypes A positive, B negative that are different from A positive, B positive as the parents, or the recipient A negative, B negative, but they're having the recombination of A positive, B negative. Under the specialized transduction, this is the bacterial DNA and this is prophage DNA. Bacterial cell has prophage integrated between gene A and B. This is A positive and this is B positive. Now, once bacterial cell has prophage integrated between gene A and B, then what will happen? Prophage DNA exits incorrectly, taking adjoining bacterial DNA with it. There is some confusion between the prophage DNA and the bacterial DNA, and they are just exist incorrectly and taking adjoining bacterial DNA within it. Then what will
Excuse me,
happen?
ma'am.
Yes.
Ma'am, somebody is asking to explain transduction again, ma'am. Please, she is requesting.
Sure.
Okay, ma'am.
Transduction is the process where the transfer of bacterial DNA from one bacterial cell to another by means of a bacteriophage. What is bacteriophage? We have the two important methods, that is general transduction and specialized transduction. Under general transduction, this is the bacterial cell, this is bacterium having the two genes in the chromosome or the genetic material, A positive and B positive, and this is the place where the virus is attached on the bacterial cell, that is on the bacterial membrane with their hooks. There is the specific place, not everywhere the virus is attaching or anchoring on the surface of the bacterium. Only at a particular place they are showing their attachment. Once such type of assembly occurs, this is known as bacteriophage. Now, phage is infected bacterial cell when bacteriophage is formed. Then what will happen? Once the bacteriophage is formed, host DNA is hydrolyzed. This is the host DNA. It is black in color. It is hydrolyzed into pieces. And this is the viral DNA, that is the phage DNA. It is also undergoing the process of hydrolysis or multiplication. Then what will happen? Whatever are the DNA and the phage proteins are present, they are also making inside this bacterial cell. They are also undergoing the process of division. Once such type of division takes place, now they are ready to assemble all these parts. What are these parts? Capsid, neck, tail, and the genetic material. All are scattered, all are differentiated here in the pieces. Now these pieces are assembled in this form, in this manner in which it was infected the bacterial cell. A bacterial DNA fragment is packaged in a phage capsid. Here in the phage capsid, all the bacterial DNA is packaged. Here they are having the A positive gene in their capsid. Now they are ready for the process of crossing over or recombination. Once the process of crossing over or recombination is going on, then what will happen? Transducing phages infect new host cells where recombination, that is crossing over, can occur. Now, after the crossing over, they are ready to infect the new bacterial cells. In which manner? In the same manner in which they have already infected the bacterial cell. Whence they are infecting the new bacteria, that is known as recombinant bacteria. Why recombinant? Because here the process of crossing over takes place between the new host cell and the virion, which is already having their capsid containing the genetic material. Hence, they are having the recombinants which are having the genotypes A positive, B negative, which is different from either from the parent or that is donor, either from the parent, donor, or from the recipient, which is having A negative, B negative. Means process of recombination is successful and we are getting A positive and B positive genes. That bacteria is known as recombinant bacteria. Is it clear? Now, under the specialized transduction, here we have the bacterial DNA. Bacterial DNA is having the prophage. This bacterial DNA contains two genes, A positive, B positive. Right? This bacterial cell has prophage integrated between gene A and B. Then prophage DNA exits incorrectly, taking adjoining bacterial DNA with it. When such type of process is going on, integration between gene A and B, by mistake prophage A is incorrectly joined with the bacterial DNA. Then what will happen? Phage particles carry bacterial DNA. Here they are containing the A positive gene along with the phage DNA. Right? Phage particles, they are carrying the bacterial DNA along with the gene A along with phage DNA. Then what will happen? Again the same crossing over process of recombination takes place. Once the transducing phages infect host cells, then what will happen? Where recombination, that is crossing over occurs, and again here we just see there is a difference between this bacterium and the first one. They are having the recombinant bacteria, that is A positive and B negative, which are different from their parent A positive, B positive as a donor, and A negative, B negative as the recipient. It means the process of recombination takes place. One character is coming from the parent side, that is donor side, and another character is coming from the recipient side. That's why the recombinant bacteria is formed. Is it clear? I think I tried my level best to clear it. Now, another category of genetic engineering, that is when we are improving the strain with the help of genetic engineering as per the standard procedure. 5 important steps of the recombinant DNA technology, that is selection of the DNA fragment, number 1, selection of the suitable vector, number 2, with the help of restriction enzymes, either endonuclease or exonuclease, then the selection of the host for the transformation, incorporation of the selected DNA fragment. Here we have selected the DNA fragment of insulin production that are inserted into the plasmid, which we have selected on the basis of the size of that insert, which is ligating in the suitable vector, which is nicked with the help of suitable restriction enzymes. Then engineered plasmid is inserted into the new bacterium for their amplification. Here, the process of amplification takes place, and what will happen? These bacterium divides and begins producing insulin. These are the methods under where the foreign DNA is incorporated and helping in the formation, helping in the process of DNA transmission, or characters of the DNA should be transmitted with the help of foreign DNA by the means of conjugation, transformation, transduction, generalized transduction, specialized transduction, and with the help of another technique, that is genetic engineering based on recombinant DNA technology. Apart from that techniques, we have some other techniques which are known as novel genetic techniques. Under this novel genetic techniques, 2 important methods are there, that is metabolic engineering and genome shuffling. As its name suggests, metabolic engineering, that is metabolism of modification, manipulation of existing pathways which are modified or entirely new ones introduced through the manipulation of the genes. Number 1, or we are using the metabolic engineering to improve the yields of the microbial product. Number 2, or to eliminate or reduce undesirable side products. Number 3. Number 4, shift to the production of an entirely new product. These are the 4 important purposes for which the metabolic engineering works. For instance, value-added products we want to produce with the help of metabolic engineering, we can produce that. Here, if we want to produce the glucose, we are producing with the help of metabolic engineering cell, amino acids, vitamins, other products, that is industrial chemicals, chemical building blocks, etc., etc., can be produced with the help of metabolic engineering. In simple words, we can say that engineering in the metabolism of any of the microbes or any of the organisms as per our desired one, as per the objective of our experiment. What are the various applications of metabolic engineering? Some important ones we are representing here. They are mainly used to overproduce the amino acid isoleucine in Corynebacterium glutamycum, Corynebacterium glutamycum, and employed to introduce the genes for utilizing lactose into Corynebacterium glutamycum to utilize whey, which is plentiful and cheap. Another application of metabolic engineering is The strains of E. coli has been engineered for the production of lycopene amino acids and alcohols. Another one is improvement of Saccharomyces cerevisiae for the production of suitable product. Now, the another category of novel strategies under the gene or the genomic uh, methods, that is genomic shuffling. Novel strategies of gene testing or gene manipulation is the genomic shuffling. The purpose of genomic shuffling, as its name suggests, shuffling from one point or one fragment to another. With shuffling of what? Shuffling of the DNA fragments. The purpose behind this is the yield of biochemically products such as ethanol and bioinsecticides is successfully improved by genomic shuffling. Here, the initial strain of the bacterium generating variant, these are the parental library of generating variants of these three kinds with the help of mutagenesis or the direct selection or the wild strain or the strains improved by rational method. Now, applications of strain selection. Applications of strain selection is basically the purpose behind this is to bridge basic knowledge and industrial application. Some important applications which are based on the basic knowledge and industrial applications are to improve thermotolerance and ethanol tolerance in Saccharomyces cerevisiae, discovering new microbial compounds and improving the synthesis of known ones, particularly amylase, protease. These are the examples. Another one is tremendous increase in fermentation productivity and resulting decrease in cultivation costs. One more application under this category is recombinant DNA technology has also been applied in medicine, particularly insulin production, HGH, hepatitis B vaccine, etc., etc. Now, summarizing this session, that is the first session is based on the microbial strain selection, their improvement techniques. Now we are summarizing this session in the form of what we have discussed. We have discussed the three important things: strain selection, on what basis we are selecting the strains to improve the technological properties, what are the various sources from where we are getting the strains or procuring the strains, that is from the already the repositories available within the nation or outside the nation. Then strain improvement to enhance their metabolic capabilities, and screening that is based on random screening and relative screening, that is absolute screening, and strain manipulation not involving foreign DNA, that is in the form of mutagenesis, site mutation, site directed mutagenesis, point mutation, frame shift, mismatch, etc., etc. And DNA involvement of the foreign DNA for the transformation or for the transmission of characters from one donor to recipient, that is in the form of conjugation, transformation, transduction, that is generalized transduction, specialized transduction, genetic engineering techniques based on recombinant DNA technology, other specialized, that is novel therapies, that is metabolic engineering and genomic shuffling. With all these, now we are summarizing this session. Now, the session is open for your kind queries, satisfaction of the kind, satisfying the kind, your kind queries and your answers. Sorry, or your questions which we are going to resolve. Right? Kindly ask. If anybody wants to ask anything, kindly put up your queries. We try to resolve them.
Ma'am, please look for the queries in the chat box, please.
Dr. Ashok, kindly go through the queries one by one. Dr. Suresh, do you hear me?
Yes, ma'am. Yes, ma'am. Yes, ma'am.
Ma'am, somebody is asking that most of the antibiotics are obtained from microorganisms. These antibiotics leads to resistance during treatment. Then what are the alternatives to antibiotics obtained from microorganisms?
You're asking about the resistance of the resistance of the microbes during the production of antibiotics. Am I right?
No, about the alternative to antibiotics. I think you
Yes,
have already mentioned in your presentation about bacteriocins.
exactly.
So, yeah, bacteriocin is one of the important alternative.
Alternative. Absolutely. Right? Any other query?
Ma'am, Kuldeep Kumar is asking why UV rays are mutagens.
The range of UV rays, as you know very well, that if you are just exposing yourself, not the microbes, you just expose around 12 o'clock during the sunshine, hardly for a period of 5 to 10 minutes. The range of that UV light is too much. That is not only acting as the mutagen in the case of microbes, but that is also in the case of human beings also. Only because of that, the range of that mutagens, whatever the mobiles, whatever the other, that is the software materials we are using, devices we are using, one way or the other way, they are also having such type of radiations.
Okay, satisfied? There's any another query?
Any query by anybody else? Why UV rays? One more important reason behind this is whenever we are using the laminar air hood, we are just sterilizing our laminar air hood with the help of UV rays. And if you want to just give the exposure to the to the any microorganism for a fraction of second, the mutation occurs. It means the range of that UV rays is too much to disrupt the normal function of that organism. Anybody else wants to ask anything?
Ma'am, one question is, genome shuffling is a shuffling of genes in chromosomes.
Genome shuffling is the shuffling of fragments of DNA. Fragments of DNA at the time of recombination, when one fragment is joined with another fragment of other DNA. That is purely based on the technique of recombination. How many places that DNA is forming the crossing over process? That fragments are joined together and they are showing the process of genomic, that is, we can say that DNA shuffling, and that occurs in the genome, then they are known as the genomic shuffling. Any any other question?
How
Any other query?
site-directed mutagenesis can be performed? This you have already mentioned.
Yes.
Yeah.
Site directed, as its name suggests, site directed, that is at a particular point, at a particular site. That is, we have already fixed if this is the point where we want to cause the mutation. We have taken one example, that is uracil deglycosidase deficient strain. Our purpose is just to change whatever are the T's. It means our target site is T. This T is converted into U. whatever is the T's present in that double-stranded DNA, all T's are converted into the U, and that double-stranded DNA is converted into the single-stranded. Likewise, here, excuse me, this is the point where the frame shift, this is the point mutation. Here also, this is the point mutation, this is the point mutation, and here, this is converted, U is converted into A. This is the point mutation. That all are included under the category of mutagenesis, that is site-directed mutagenesis. Any other query? Anybody wants to
Ma'am,
ask?
another question is, at what level can CRISPR-Cas9 be used for strain development? At what level can CRISPR-Cas9
Yes.
be used for strain development?
If you are dealing with the RNA, mainly we are using the CRISPR-Cas for the RNA editing, that is gene manipulation, right? If you want to use this is the latest technology nowadays, that is CRISPR-Cas9. One of our students did this, Minu Sachdeva used this tool for the alteration of the level of methylation in the lung cancer cells, right? If we want to just improve the quality of the strain with the help of CRISPR-Cas9, we can do that successfully. But your motto should be clear. For which purpose you are improving your strains? For which purpose you are developing your strain? CRISPR-Cas9 is a wonderful tool as the editing tool nowadays to just manipulate the gene at any of the levels. Any other query?
No, ma'am.
Sure.
Yes, ma'am. In chat box, no.
Anybody wants to ask anything? Shall we proceed to the another session of today's workshop? In this manner, we have end our first session that is based on the microbial strain selection and their improvement techniques. Now, beginning of the second session, that is based on media preparation, broth preparation, selection of the media, and all, and many more things we have to develop, we have to discuss in the second session. For this purpose, I'm inviting Ms. Shivangi Srivastava, MTech fourth semester student of School of Biotechnology, RGPV Bhopal, to discuss the various features of media preparation and their selection. Shivangi Srivastava.
Thank you so much, ma'am, for inviting me to discuss about the media preparation, selection, and broth preparation. I am here to discuss why media. Tell me why media is needed for microorganism growth, because media is just a food. So. It is the basic requirement of microorganisms or sometimes cells for converting large molecules into ATP for their growth and survival is known as media. We are simply saying like media is a food for microorganisms which are rich in nutrients, minerals, macromolecules, vitamins, macroelements, etc. etc. So the basic component of any media is uh, the macro elements which are required more than 100 mg for any microbial colony. They are like sulfur, nitrogen, phosphorus, potassium, magnesium, calcium, and microelements that are required less in amount and but they need for their metabolic reactions like calcium, nickel, ferrous, zinc, etc. etc. And they use sometimes minerals and vitamins, and they need amino acids too for their synthesis of protein and for their synthesis of any other uh, process. So there are three types of media, from which one is solid media, second is semi-solid media, and third one is liquid media. So, what is solid media and why it is important? Solid media are the media which provide the area for growth of microbes either on surface or in induced it. Okay. For solidifying the media, agar is used in some proportion. Its range vary from 1.5 to 2%. The gelling agent in agar is an unbranched polysaccharides obtained from the cell walls of some species of red algae, primarily from the genera Gelidium and Gracilaria. It is widely used because it contains favorable environment for most of the microorganisms. Other advantage is that it is solidified at 45 to 50 degrees centigrade, and its melting point is near to 100 degrees centigrade. If you are going for the history of using agar, why agar is used for the solidifying agent, then I would like to introduce about Robert Koch. He is the father of culture media. His first success in bacteriology was the isolation of Bacillus anthracis, which at the time was causing the disease anthrax in the cattle. This was the first time that any pathogenic organism has been isolated and studied outside of the host body. So he postulates something two positive agents of infectious disease. He published in 1882. Then he published some postulates from which the first postulate is the organism must be consistently associated with the disease. Second one is it must be isolated and grown in pure culture. And third one is the pure culture must be shown to induce the disease when injected into experimental animals. Okay, so one day he is working. First of all, he will use urine as a media for isolating the bacteria. After some time, he saw bread. He saw a red blood spot on bread or the fermented bread. Then when they go for the further processes, how it comes and what is this, so they concluded that there are bacterial colonies. So he wanted to use the solid media. So after that they use gelatin, but the very disadvantage of gelatin.
Shivangi,
Yes, ma'am?
extremely sorry to interrupt you. Please explain slowly. Somebody's just saying, kindly explain slowly, right?
Okay, ma'am. So, can I speak again about history, or I'm?
No, you have to start from the media, right? Not the
Oh,
history.
okay, ma'am.
Start from the
Okay,
media
ma'am.
slowly and gradually. Okay?
Okay, ma'am. Okay. So, his co-worker's wife Fanny has suggested them for using agar for solidifying agent. So, from that time, we are using solid agar for solidifying the media. So after that, semi-solid media. What is that? Semi-solid media is a media that contains less amount of agar, and it is like custard-like consistency, and it is used for the cultivation of microaerophilic bacteria or for the determination of bacterial motility. If you isolated bacteria, but you want to check the motility of bacteria, then how can you check? So you will be check from using semi-solid media. What is liquid media? Liquid media is natural broth, is also known as broth media, and broth is purely liquid without agar, and it is transparent. But if there is a growth occurs, then it will be turns from transparent to translucent because growth of microorganisms show in the form of turbidity. So it is used for inoculum preparation in upstreaming process of industries like probiotics. Some industries produces probiotics, so they are using liquid media as inoculum preparation for that in upstreaming processes. Here are some outcomes related to media using media. Solid media and liquid media is used at a time for cultivation of microorganism, because when you are going to isolate a microorganism from an uh, unknown sample, so you are first of all you use liquid media and inoculate it in liquid media. Turbidity came. After that you further go for serial dilution, and after that you pour that microbial colonies on the microbial sample on the solid media. After 24 to 36 minutes you can calculate the colonies of that microorganism. After the and as per the requirement, media could be prepared. Solid media and semi-solid media preparation is quite different from liquid media, because there are some agar present in the solid and semi-solid media, but liquid media have nothing agar. So how to prepare a solid media? So there is a preparation method. Firstly, dissolve the water into agar by adding total volume of distilled water. You should mention this in your mind, that in broth you should use 2/3 part of the water while dissolving the media, and for solid media you should add the agar in the total volume. Placed it in on hot plate for proper mixing, because agar is not mixed at normal temperature, so it needed some high temperature for proper mixing. When it totally mix, it will show the transparency. You can transparency, and you can see the bubble from bottom to upside. Bubbles come from bottom to upside. So transfer it into two conical flasks. You uh, you placed uh, you placed the you use the agar which was on using beaker, because when you put it on hot plate, then beaker is very reliable to you for mixing, proper mixing. So for if you are going for further process, like if you are going to sterilize the media, beaker is not that much applicable for your sterility. So you should change your flask from beaker to conical flask. After that, sterilize it at 121 degrees centigrade and 15 lbs pressure for 15 minutes. Then pour it when temperature degrades up to 50 degrees centigrade. How can you check the temperature degradation? You can check whenever your media is cooling. You can check with putting on your vest. If you are putting on wrist, uh, if it is applicable to you, okay, now you can use for pouring in the plates. Why it is important and why it is used? It is used for colony morphology, pigmentation, and hemolysis. How to prepare semi-solid media? Semi-solid media is this process is same like solid media, but here is difference in agar content. So it will take less time for making it. Used for checking motility of microorganism, as I told you before. After that, how to prepare liquid broth? First of all, take two-third part of the distilled water in your conical flask of total volume. Now add the powder media into conical flask. Mix it with the use of magnetic stirrer and by simply from hands by shaking. After mixing, now calculate the amount. How many before this process you should check the pH of that broth, because if you will check after the makeuping on total amount of media, then it will change your media volume too. So you should check before adding water. You should check pH before adding water. Now calculate the amount with the use of measuring cylinder. How much amount you needed to complete your total volume. So add water up to make up to total volume. After that, use cotton plug for covering the media, because cotton plug is used for removing the contaminants entering in the media. And autoclave it at 121 degrees centigrade temperature and 15 lbs pressure for 15 minutes. Now store it as stock, and whenever you use, you have a stock for it. If you are going to use right now, then you can use that after cooling down up to 45 to 40 to 45 degrees centigrade. Otherwise your microbial sample will degrade. Uh, Here are the selection of media. Why we are using the selection of media? Because selection of media is important if you are going to work on different microorganisms. Different microorganisms need different molecules for their growth, or different macro elements for their growth. So on the basis of requirement, different media is used. There are different type of media available nowadays in the market. for differently microorganisms growth. Uh, Used for inoculum preparation, blood culture, continuous culture. There are four types of media which are using nowadays, like simple media. Simple media is the media with basic requirement of microbes, also known as vessel media. And what is vessel media? The vessel media is are those that may be used for growth or culture of bacteria that do not need enrichment of the media, like nutrient broth, nutrient agar, peptone water, and these are used for the isolation of staphylococcus and enterobacteriaceae grown in these media. Okay, so this is normal media for growth of almost all type of microorganism. Nutrient agar having the nutrient broth and agar having only the difference of agar in the constituents, like peptone, meat extract, and NaCl is the basic component of nutrient broth, and for agar it contains 2% of agar. After that, complex media. What is complex media? Complex media usually contain complex materials of biological origin, such as blood or milk or yeast extract or beef extract, or the exact chemical composition of which is obviously undetermined. If you can't understand this, so I have other definition for complex media too. It's the combination of extracts and sugar creates a medium which is rich in minerals and organic nutrients. But since the exact composition is unknown, the medium is called complex. Uh, Here is the best example for complex media is plasma, blood plasma. Blood plasma is used, but no one knows the composition, and no one knows the exact amount of that composition. So this is also come in complex media. After that, synthetic or defined media. A defined medium, also known as chemically defined medium or synthetic medium, is a medium in which all the chemicals used are known. No yeast, animal or plant tissue is present. The term chemically defined media is often misused in the literature to refer to serum albumin containing media. There are examples. Peptone water contains 1% peptone plus 0.5% NaCl, which is used to test the indole formation. After synthetic media, there is special media. Last one. The special media, or having five types: special media, selective and differential media are from which selective and differential media are used to isolate or identify a particular organism. Selective media allow certain type of organisms to grow and inhibits growth of other organisms. So special media is of five types. Here are the types of special media. Firstly, the enriched media. Second, selective. Third, anaerobic, transport, and differential media. So we are going to talk about different type of media in short. So enriched media, the media are enriched usually by adding blood, serum, or egg. Example: enriched media are blood agar and chocolate agar. Selective media, these media favor the growth of a particular bacterium by inhibiting the growth of undesired bacteria and allowing growth of desirable bacteria. Inhibitors that are added to it are antibiotics,
Shivangi, very sorry to interrupt you.
yes,
Shivangi,
yes sir.
please go slowly. Please go slowly. है ना?
Okay sir.
You are too fast. The candidates are asking to explain slowly. Okay?
Okay sir. So can I use
No, no,
sir?
from here.
Oh, okay, sir. Okay. What is enriched media? Enriched media containing the enrichment of any sources, like if microorganism need something for their growth. So by in this media, we are adding blood in the time of pouring in the plates. So this is enriched media. Okay, selective media. These media favor the growth of a particular bacterium. If you want to grow or isolate a microorganism from n number of microorganisms, so on that time, a particular media is available in the market For a particular bacterium, you are using a particular media for that, for the bacteria that you want to grow. Okay, so there are some inhibitors in that media which slow down, or which are inhibiting the growth of undesired bacteria, or allowing growth of desired bacteria. So there are some inhibitors mentioned here, like they added antibiotics, dyes, chemicals, or alteration of pH. There are some examples. First example show the tellurite media. The tellurite media inhibits the growth of most of the thought organisms except Diphtheria bacilli, because this media is used for the isolation of Diphtheria bacilli. Iosine methylene blue agar. It is used for the growth of gram-negative bacteria, which inhibit the growth of gram-positive bacteria. Campylobacter agar and Lowenstein-Johnson medium. These are also came in the selected media. What is differential media? In differential media, an indicator is included in the medium. A particular organism causes change in the indicator. It's like if you are going to stay in a microbial, like if you are going to stay in a microbial, you want to grow gram negative bacteria. So indicator show the color changes in that. Like there are some how can I explain? Okay, I will explain this from Clade and XLD. Clade is the agar. XLD is the xylose lysine deoxychioate. It is used to isolate Salmonella and Shigella species from stool specimen. Okay, or Clade is used. They are using because they just inhibit the if some microorganism inhibiting the lactose. Okay, so if it will not use the lactose for that, so it will change that color. If some of them are using, then it will not changing that color. As like these are differential media. This can differentiate your particular organism from the n number of colonies. Example of Meconi agar as indicator media. Clade agar is used for the isolation of organisms from urine, and XLDE is used for stool specimen. So this is transport media. These media are used when a specimen cannot be cultured soon after collection. So when you are going to order a microbial strain, okay, so they just put that in agar or in something which have to be maintain their motility. Okay, so they use transport media because you are ordering from Imtech Chandigarh, then it will come to Bhopal. So you are not going for cultured after collection, so it will be like stock. Okay, stock for you. So transport media is used in that time. For example, Carey Blair medium, amines medium, and Stewart medium are used for transport media. In last, anaerobic media. What is anaerobic media? Anaerobic media only grows or only cultivate the anaerobic microbes. It inhibits the oxygen content from the environment as well as from the media. So it maintains the anaerobicity throughout the growth. So this media is used. And there are some examples: Robertson's cooked meat medium and thioglycolate broth medium. These are used for anaerobic bacteria cultivation. Here are now precautions. What precautions should be taken while preparing the media? So whenever you weigh the powder form of medium or something extra that you want to weigh with that, like if you want to weigh yeast extract along with your media, so always check the calibration, because the weighing machine is very sensitive to air. If you are talking on that time, if you are sneaking on that time, it will change or it will fluctuate your readings of calculating. So calibration should be checked properly. At hot plate, agar should be stirred continuously, otherwise it would be burned or stick on the sides of the beaker. So you should maintain that you have to stir continuously your agar until until your agar will becomes transparent. Temperature should be checked before pouring in the plate, because it will change the order of agar. If you are pouring very hot in petri plates, then it will be it will change the surface of agar, because sometimes it will be like very high surface, and sometimes here will be like very low surface. So pouring time will be on when your temperature degrades up to 50%, just before it's solidifying. Check the pH of broth before make up the media with distilled water. Why? Because if your pH fluctuate from if you want pH of 7.8, but it will become, after adding all things, it will become 8.8. So you should check and you should calibrate that. So you will add some acidic like HCl, so it will also change your volume of media. So before makeuping, you should check the pH of broth. Cotton plug is effectively airtight, otherwise contamination would occur. If your cotton plug is not airtight, then it will grow unwanted colonies, and so many unknown colonies also grow on that. Now there are some precautions which we have to taken. Like, products are to be used only by adequately trained and qualified laboratory personnel. Observe approved biohazard precautions and aseptic conditions and aseptic techniques. Care should be taken to avoid contact with the skin, eyes, or mucous membranes when handling culture media or any laboratory reagent, stain, fixation, or chemical. And the last and most important thing, glassware should be sterilized before using the preparation of media. Thank you. Now I would like to invite Dr. Mrityunjay, Assistant Professor of the School of Biotechnology, for discussing about the serial dilution. Now, thank you everyone.
Nice presentation, Shivangi, and thanks for your kind words.
My pleasure, sir.
Now, good evening, everybody. Now we are discussing serial dilutions. What is serial dilution? Serial means repeatedly or periodically. Dilution is a process of decreasing the concentration of a solute in a solution, usually simply by mixing the more solvent, like adding more water to the solution. To dilute a solution means to add more solvent without the addition of more solute. This picture shows shows concentrated solutions where more solute particles per unit volume, and this shows diluted solution where fewer solute particles per unit volume. Moles of solute before the dilution is equal to moles of solute after the dilution, means the number of moles of solute during the dilution process is not changed. Many laboratory chemicals such as acid are purchased as concentrated solutions, that's known as stock solutions. More diluted solutions are prepared by taking a certain quantity of stock solutions and adding it to the diluent, which is typically water or buffer. Dilution factor is the total of the unit volume in which your stock solution will be dissolved. Dilution factors refers to the ratio of volume of the initial concentrated stock solutions to the volume of the final diluted solutions. This is V1 to V2. Dilution factor can be calculated, which is equal to V2 divided by V1. Serial dilution. It is stepwise dilution of a substance in a solution. The first step is making a serial dilution is to take a known volume, which is usually 1 ml of a stock, and place it into volume of distilled water, usually taking 9 ml. This is called tenfold dilutions. Dilutions in serial dilutions you can use by twofold, threefold, fivefold, tenfold, or hundredfold dilutions, where initial volume is most of the cases is 1 ml, and final volume if 2 ml it's twofold, if 5 ml it's fivefold, and if 10 ml it's tenfold. Likewise, if for 100 fold solutions, if you are taking 10 microliter of a stock solutions and adding it in 990 microliter of solvent and making 1 ml of final diluted solutions, its 100 fold dilutions occur. This produces this one is produces 10 ml of dilute solutions. This dilute solutions has 1 ml of extract per 10 ml, producing a tenfold dilution. That is, amount of stock in each ml of the diluted solution is 0.1 ml. Serial dilution is used in experimental biology and in highly diluted solutions, and in case of determining the concentration of microscopic organisms, number of cells, and in isolation of microorganisms. Many studies require the quantitative determination of bacterial populations. The two most widely used methods for determining bacterial numbers are the standard K method, and second one is a spectrophotometer analysis, that is turbidimetric analysis. The plate count method is an indirect measurement of cell density, live bacteria, because you are counting the colonies which is formed by live cells. That spectrophotometer analysis is based on turbidity and indirectly measures all bacteria, means cell mass of all bacteria which are dead or alive. The number of bacteria in a given sample is usually too great to be counted. However, if the sample is serially diluted and then plated out on an agar surface in such a manner, the single isolated bacteria form visible isolated colonies. The number of colonies can be used as a measure of the number of visible living cells that known dilutions in the known dilutions. Keep in mind that if the organism normally forms multiple cell arrangements such as chain, the colony forming unit may consist of a chain of bacteria rather than single bacteria. In addition, some of the bacteria may be clumped together. Therefore, when doing the plate count technique, we generally says we are determining number of colony forming units in that known solutions or in that solutions, not bacterial numbers, bacterial cell numbers. By extrapolation, this number can be used to calculate the number of CFU in the original samples. Normally, the bacterial sample is diluted by factor of 10 and plated on agar. After incubation, the number of colonies on the dilution plate showing between 30 to 300 colonies is determined. A plate having 30 to 300 colonies is chosen because the range is considered statistically significant. If there are less than 30 colonies on the plate, a small error in the dilution techniques or presence of a few contaminants will have a drastic effect on final count. That called TFTC, means too few to count. Likewise, If there are more than 300 colonies on the plate, there will be poor isolation and colonies will be grown together. That called TMTC, means too numerous to count, or TMTC means too many to count. For a more accurate count, it is advisable to plate each dilution in a duplicate or triplicate and then find an average count. At the end of the incubation period, count by looking at the bottom of the plate while keeping the petri plate closed. Agar is translucent, so it should not have to open the plate. Select all the petri plate containing between 30 to 300 colonies. Count the colonies on each plate. If there are lot of colonies on the plate, helpful to use a marker to mark the colonies already counted. Now, colony forming units. Not the same as bacteria because two bacteria might have been very close and form one colony. Calculate the number of bacteria here. For that, we are using colony forming units per ml or gram of samples by dividing the number of colonies by the dilution factors multiplied by amount of the specimen added to the agar plate. That is CFU per ml of samples, number of colonies divided by amount plated multiplied by dilution. Examples here, if you are having 46 colonies on your plate, you put 1 ml of bacterial culture into 99 ml of saline and plated zero point 0.1 ml of it on agar plate. Dilution factor is 1 by 100. CFU per ml of samples. Number of colonies 46. Amount plated 0.1 ml and dilution is 1 by 100. After calculating, you find out your result, that is 46,000 CFUs in that. Now, dilution series or series of dilution. Here, this stock solution is diluted in three different ways. In first case, 1 ml of stock solution is added in 9 ml of diluent. Total volume is 10 ml. Dilution is 1 by 10 and final dilution factor is 1 by 10 for tube 1. In second case, 0.2 ml of stock solution taken and add up in 9.8 ml diluent. Total volume of the solution is 10 ml. You calculate the dilutions by 0.2 ml divided by, initially you take 0.2 ml and final solution volume is 10 divided by 10 and it simplify in 1 by 50. And in third case, you are taking 0.1 ml of stock solutions, adding in 9.9 ml diluent. Total volume is 10 ml. You calculate the dilution factors by dividing initial volume by final solution volumes, 0.1 ml initial and 10 ml final. That simplifying 1 by 100. Now, serial dilution. Same dilution factors at each stage. This picture shows twofold serial dilutions. Here, stock solutions is diluted by twofold. Taking 1 ml of stock solution, adding in 1 ml of diluent is total volume is 2 ml. Dilution is initial taking volume is 1 ml, total volume 2 ml, dilution factor is 1 by 2. In second stage, 1 ml of this dilution samples are taken from tube 1 and adding it in tube 2 where 1 ml of diluent contained. Total volume is 2 ml and dilution is 1 by 2 with respect to tube 1, but final dilution factors in respect to stock solutions. For that, you just multiply these dilution factors here 1 by 2 multiply by 1 by 2 is equal to 1 by 4. For tube D3, you are using 1 ml of solutions taken from tube D2 and adding in 1 ml of diluent. Total volume is 2 ml. You are just 1 by 2 diluted in this step. Final dilution factor is just multiply 1 by 2 into 1 by 4, that is 1 by 8. Means tube D3 contain 8-fold total dilutions of stock solutions. This picture shows 10-fold serial dilutions. Here, stock solutions, from stock solutions you are taking 1 ml and adding 9 ml of diluent, and likewise in 5 different steps. Each tube contains total volume 10 ml and 1 ml from initial tubes or earlier tubes. Each steps having dilutions 1 by 10, and in respect to stock, the final dilution factors for tube D1 is 1 by 10. For tube D2 is just multiply 1 by 10, 1 by 10, 1 by 100. For tube D3, multiply by 1 by 100 into 1 by 10, that 1 by 1000. And for tube D4, it's 1 by 1000 multiply by 1 by 10 is 1 by 10,000. And for tube D5, it's 1 by 10 multiplied by 1 by 10,000 is 1 by 1 lakh. Means tube D5 contains 1 lakh fold total dilutions in comparison with stock solutions. Here, we can calculate CFUs per ml in serial dilutions experiments. Here, stock solutions contains, that is initial solutions, 1000 CFUs. Taking 1 ml and adding 9 ml of diluent means total volume is formed 10 ml and dilution is 1 by 10. So just divided this CFUs by 10 are using dilution factors at what is there. Here it's 10 times. So just divide this and you get your CFUs per ml in diluent solutions. That 1000 divided by 10 is 100 CFUs per ml. And this one is other example. Here initial samples having 35,000 CFUs. Just here also the dilution factor is 1 by 10 at each step. So just divided by 10 and you get your CFUs per ml in that tubes. Here tube D1 contains 3500 CFUs per ml because 35,000 divided by 10 is 3500. Tube D2 contains 350 because 3500 divided by 10 is 350 CFUs per ml. And tube D3 contains 35 CFU because 350 divided by 10 is 35 CFU. And this types of experiments you just did because you put that in the range of 30 to 3000 CFUs so that you choose that one. Viable count assays. Here, a sample is diluted by serial dilutions where 1 ml of sample is adding in 9 ml of water and repeated for 6 times. Means you are diluting the sample by 1 by 10 factors and plated 1 ml of these diluted samples on plate and incubate for overnight. After that, you get the first 2 tubes having so many number of colonies are overlapped called T and TC, means too numerous to count. And third tube contain, respect to third tube, the plate is having 250 colonies count, and fourth one is 23, fifth is 2, and sixth is having no colonies on plate. So 23 and 2 is less than 30, so you are not using this for calculation of CFUs in this unknown sample. Here 250 is in between 30 to 300, so you are using third one. Here just multiply by amount plated, which is 1 ml in this case, and dilution factor per tube third it's 1000. You get 2.5 into 10 to power 5 CFUs per ml. If you know the total amount of sample, you just multiply with this and you get that total CFUs in your sample. This is second example. Here you just take original inoculum sample and dilute it serially, and here you get in fourth tube, plate of fourth tube where first 3 is having too many colonies, fourth having 32 colonies on plate, and fifth is less than 30. So you just yes. So Here, dilution factor is 1 by
Your
10,000.
Patil, please mute your mic. Please mute your mic.
Hello.
Please carry on. Carry on.
Yes, sir, please continue.
Just multiply by 10,000, 32 number, and you get 3,20,000 CFUs per ml. And if you know the original quantity of the sample, you just multiply with that and you find out total number of CFUs in that samples. Thank you. If any queries, just write down in chat box. We'll entertain in at the evening, at the end of this sessions. Now, I want to invite Riddhi Tiwari for next presentation. She is PhD student in School of Biotechnology, RGPV Bhopal. Over to you, Riddhi.
Thank you so much, sir, for introducing me. And a very good evening to all the participants. Thank you for showing your interest in our second workshop of the Consortium Biotech workshop. For now, I am going to present my presentation regarding which will be the concluding for presentation. So the topic of this presentation is preparation of agar plates. Till now, we have known how to prepare, how to select a strain. Then for how we are going to grow that strain, for that we have known that media is required, and how we are going to see the growth of the bacteria or any microorganism. For that we have known about the serial dilution. Now we require a solid base also, or semi-solid base on which we can grow our bacteria or our microorganisms. For that we require our agar plates. So let's know something about the preparation of agar plates. Before we go through the process of preparation of agar plate, we should know that a petri dish and an agar plate, there is a difference between petri dish and an agar plate. Petri dish is nothing but an empty vessel which is made up of glass or plastic, and it derives its name from the inventor who invented this vessel. When it is empty, it is called the petri dish, and when we pour our required media into it, then it becomes agar plate. So we can say that agar plate is a petri dish that contains agar as a solid growth medium along with few nutrients, and it is used to culture microorganisms. Agar is commonly used in the laboratory for the growth of the bacteria and other microorganisms because it acts as a food for them. It acts as a culture that provides nutrients and a place for them to grow. The advantage of this is that it is indigestible to the microorganisms, and therefore they are not able to eat it or destroy it. Some of the commonly used types of agar in the laboratory routinely in microbiology laboratory are McConkey agar, chocolate agar, tryptic soy agar, triple sugar iron agar, hectoine agar, and mannitol salt agar. And the most commonly used and the basic agar which is used to grow microorganisms is nutrient agar. The different colors depicting here are the colors obtained while after the preparation of the media. Like so, McConkey agar will always be red in color, and hectoine agar will always be in green color. So that's why I have specified them with particular colors. So we have known about the introduction of what an agar plate is. Then let's know about the steps that needs to be followed while preparing our agar plate. The first and the most important thing is we should sterilize all instruments, solutions, and media before we use them for plating procedures. Next is we should clear away all the clutter around our working area so that there is no disturbance, no hindrance. Next comes the cleaning of the workbench. We should clean it properly and disinfect it with a commonly used disinfectant like 70% ethanol so as to remove any possible contamination. After this, we should set up a Bunsen burner, and while working with Bunsen burner, we should always work slowly, carefully, and deliberately. An important point to note here is that when you are working with biosafety level 2 microorganisms, you should set up your work or you should carry on your work in a biosafety cabinet. But here, a Bunsen burner cannot be used. Why is it so? Because the flame of the Bunsen burner will disrupt the airflow, and that airflow we all know is the most essential component of the proper functionality of this laminar airflow hood or a biosafety cabinet. Next, what we need to do is we need to label our Petri plates very carefully using colored marker pens that indicates the type of the medium which is being poured, type of the microorganism either added or later on we are going to streak on that. If we have added any antibiotic like ampicillin or tetracycline, the date of the preparation, your name, as well as any other necessary details. So we should always label our Petri plates carefully and clearly. Otherwise, it can lead to a lot of confusion and it can hamper the ongoing experiment. After the pre-preparation of what we do before pouring our media in the Petri plates, next comes how we are going to do that. For that, we need to follow the following steps. First of all, we need to collect a bottle or a flask containing sterile and molten agar. Next, hold the bottle in the right hand, remove the cap with the little finger of the left hand, flame the neck of the bottle, then lift the lid of the Petri dish slightly and pour the sterile molten agar into the Petri dish and replace the lid. You can continue this process for the required number of Petri plates that you need. After pouring your media completely in all the Petri plates, next flame the neck and replace the cap. It is always advisable that you use all the media that has been autoclaved and prepared, and if not, you can still replace the lid and store it for later use. Next, what we need to do is we should gently rotate the dish so as to ensure that the medium is completely spread in the Petri plate. Now allow the plate to solidify. It is also advised that you should use the plates immediately or as soon as possible after the pouring of the media. But if it's so that you don't want to use it immediately and you want to store them, then it is advised to tape them with paraffin, store inside the sealed plastic bags, and refrigerate them at 40 degrees centigrades so as to prevent the agar from drying out. This was the basic method of preparing an agar plate. Next comes another aspect of preparation of agar plates, that is pour plate method. As we have already seen in the previous presentation by Dr. Mrityunjay, that how we prepare cereal dilutions. So it is a continuation of this. So pour plate method is the method that is used for counting the number of colony forming bacteria present in a liquid specimen or a sample. For this, A fixed amount of inoculum from a broth or a sample, usually 1 ml of sample is taken after serial dilution, is placed in the center of sterile petri dish using a sterile pipette. I am everywhere using the word sterile because we have to ensure that whatever we do it is in aseptic conditions, about which we have earlier spoken about in our earlier workshop. Next, what we need to do is we have to pour the sterile molten agar, which is near about 15 ml to 20 ml as per your requirement. It is then poured into the petri dish above the inoculum and mix it well. After the solidification of the agar, we need to invert the plate and incubate at 37 degrees centigrades for 24 to 48 hours. Now, in this case, microorganisms can grow both on the surface as well as within the medium. The difference between the colonies that grow above the medium and within the medium is that the colonies that grow within the medium are usually smaller in size and might be confluent, but the few that grow on the agar surface usually are of the same size and appearance as we obtain after a streak plate method or a spread plate method. Next, after doing this, each colony is carefully counted using magnifying colony counter. Each colony represents as a colony forming unit or CFU. So this is the figure on the left hand side depicts the method of serial dilution. Like we have taken 1 ml from the, first of all, we will fill up all our tubes with 9 ml of our broth. Then we will add 1 ml of original inoculum in the first tube. That will give us a dilution of 1:10. From this 10 ml, we are going to take 1 ml. We'll first put it into the petri plate. Then we'll pour our media. And then after incubation, what we see is there are no microbial colonies, as if it's a microbial lawn. It's full of colonies and we cannot distinguish. Next, we again take 1 ml from 1:10 diluted broth and we'll add it to another 9 ml. This will give rise to 1:100. This will also give a microbial lawn as well as with 1:1000. But after taking 1 ml from 1:1000 dilution, we will get a dilution of 1:10,000, and this is the most desired one because here we are getting around 32 colonies. And if we are going to further dilute it, then the number of colonies will after certain amount of time they will not grow because there might be not enough microorganisms in that sample. So on the right hand side is the picture which I just told you about. For that, what we need to do is we have to melt our agar generally in a circulating water bath. Temperatures should lie between 44 to 46. That means it should be near about 50 degrees centigrades. Then prepare the microorganism suspension as per your requirement of your protocol. Then pipette the sample onto the bottom of the petri dish and try to make duplicates and triplicates so as to confirm our results. According to your procedure and your protocol, you have to pour near about 12 to 20 ml of molten agar over the inoculum. Then swirl the petri plate properly so as to mix the organisms throughout the agar. Once the agar has been solidified, invert the plates and place it in the incubator and incubate them as per the requirement at a temperature as per your requirement. After incubating for the desired at a desired temperature and a desired amount of time, colonies will appear. Now count the colonies and you can count the colony CFUs as well. Here we can see that colonies under the agar surface will be smaller and those at the above surface will be larger. There are a few helpful tips which will help you to troubleshoot any problems whatsoever if they come while carrying out your procedure of plating your agar plates. So it is advisable that you should pour the agar that is about near about 50 degrees centigrades. If it is much more cooler than 50 degrees centigrades, then it will tend to form bubbles and that can solidify, which can confuse you for being as if colonies. Like you can see in the figure, the bubbles usually confuse us and we think that it is a colony. So it's our utmost requirement that we should always get rid of any bubbles while doing our procedure of plating. So how to get rid of those bubbles? If you get lots of bubbles, then one of the best methods is to pass a flame of Bunsen burner over the surface of the agar plate. What will happen is the heat of the flame will help to pop up the bubbles. But make sure you do this when agar is still molten. Means it cannot be done after the solidification of the agar. Still it should be in melted form. Then only this can happen. But make sure that you may move the flame quickly over the surface of the agar. Otherwise, it is a danger of melting the petri plate. Likewise, you can also get rid of the bubbles by pricking them and bursting them with the help of sterile toothpicks or sterile microtips. As I have mentioned that for thin agar plates, like if you are going to use your petri plates immediately, then you should be making thin agar plates. For that, you should pour the agar until it has just covered the bottom of the petri plate. And if you want to store them for longer periods of time, in that case, you should pour a thicker plate, which is near about half the volume of the petri plate. Some other helpful tips that would be helpful are that most of the times we observe that there are some bubbles on the lid of the petri plate. They arise because of the condensation during the storage in the refrigerator. So to prevent condensation of water in plates, we should let them solidify and dry at room temperature until all the moisture is freed from the lids. You should store the plates upside down in the refrigerator or cold room. If they are stored in a room, check the plates after a few hours for condensation in the lid. If you find any condensation, the only trick is to turn the plates over. After after checking for condensation, you should also examine the plates for any growth of microbial colonies during the storage. That might be a major cause of contamination. Now, if your plate is having no contamination and if your plates are in a proper fashion, that means there should be no condensation, neither there should be any condensation nor their plates should be very dry. Then and then only you are able to use your plates for further procedures. Sometimes we need to add antibiotics in the plates. So in that case, medium that contains antibiotics should not be used after a few weeks of storage. For example, ampicillin is in particular will stay at its working condition for about a month if it is stored in 4 degrees centigrade in a refrigerator, and if it is kept at room temperature, then its efficiency gets lost. So this is a video that I'm going to play for you where you can understand how actually we prepare our plates. See, he is working in his biosafety cabinet and he has not used any Bunsen burner. He is pouring near about 15 to 20 mLs of media in his plates. He is stacking each plate one on the other. and immediately replacing it with its lid. So in this way, he has finished his plating. Now he will be spreading all the plates over through the cabinet. Before that, if he observes any bubbles in the media, he can just make use of the flame of the Bunsen burner, as I just mentioned, and you can just move that flame over the plates so as to get rid of the bubbles. Now he will be placing all the plates all over the cabinet and let them cool down and solidify, and this would take about 20 to 25 minutes for the plates to solidify completely. That's it. I thank you all for listening to me attentively. And now to sum up this session, in this session, the summary of this session is that in this session, we learned something about media preparation, cereal dilution, and plate preparation. For the media preparation, first of all, we should know which type of media we require for which type of microorganism that we want to grow. Once it is decided, then we prepare our media according to the protocol, or we can use the ready-made available media. Then we need to sterilize it and store the media properly as per the directives. Next, in cereal dilution, we learned that we should first of all determine the proper dilution liquid. For that, we need to prepare several test tubes with 9 ml of dilution liquid. Then we perform the first dilution. Then we perform the second dilution, so on and so forth until we reach our required dilution. Then in plate preparation, we need to first sterilize our working bench. Then we need to collect our required molten media, sterile petri dishes, and any other required supplies like marker pens, parafilm, etc. Pouring of the media in the petri dishes and storing them properly, either in the refrigerator or at room temperature. Then in this only we studied about the pour plate method. For that we should obtain the required dilution. Then we should add the desired diluted inoculum in the empty petri plate. Then we pour the molten media over the inoculum and incubate, and later on we observe the colonies by counting the CFUs. So this is all about this session. And now I would like to call upon Professor Dr. Archana Tiwari, Dean, Faculty of Applied Sciences and Director of School of Biotechnology, Rajiv Gandhi Pradyogiki Vishwavidyalaya, Bhopal, for the conclusion of this session. Thank you so much.
Thank you, Renu, for your kind words. Now the time to sum up today's session. Today we have two sessions. First session, we dealt with the microbial strain selection and their improvement, how to improve their properties, how to manipulate the properties of the microbial strains. And second session, we have dealt with the media preparation, selection of media, types of media, and cereal dilution, pour plating. We thought these sessions are very helpful for you. Whenever you will perform your experiments, you are just thinking about all these precautions which we have to take so that you will get your results in a better way. For these kind words, we are just stopping today's both the sessions, and then we will meet tomorrow at the same time on the same platform. Till then, stay home, stay safe, and feel happy. Enjoy whenever, wherever you are and whatever you are doing. Thank you, one and all. Thank you for the patience hearing. Thanks a lot. Now the meeting is going to end here. A very big thanks to all of you.